Good morning.
Good morning, sir.
Okay, fine. So let's begin. We have around 16 students on board today. So yesterday we started with the actual discussion uh, where I think so now all of you got an idea that what are primary metabolites, what are secondary metabolites. The broad classification of secondary metabolites, which I told you could be the three major classes are there. That is the alkaloid, then phenolics and terpenoids. Okay. Uh, Then we have seen the difference between primary and secondary metabolites. And then we started with a topic that what kind of challenges does these plants they have. Okay, and one of the major challenges with respect to their defense mechanism. Okay, so whenever there is a defense mechanism which we talk about, there are two types of defense mechanisms which come into picture. The first one could be your physical, and the second, the one in which we are interested, that is the chemical. So in case of physical, it could be some physical perturbations which are present onto the plant systems. So it could be with respect to the presence of the thorns, the waxy layer, etc., etc. Right. So like this, there are different physical mechanisms whereby the plant can overcome the different biotic or the abiotic factors. Then few examples which I have told you about the squad gun defense mechanism, or it could be the third party defense mechanism, etc., etc. So from that we get an idea that plants do have some specific type of the mechanisms where they can overcome these particular different factors, right? Then we started with the chemical one. So in that, I think so we started with the yeah the in this chemical defense mechanism there are two types what we saw. First one was the passive and the second was the active. So this gives us an idea that within the plant system there are some compounds which are already prepared and stored into the plants. So whenever there is an attack, these compounds could be released and the work can be done. Right now, this is I am talking about the secondary metabolites, and there are some class of the secondary metabolites which come under the active response. What does it say? That whenever there is an attack of a particular insect or a bacteria or any other kind of damage is going to be there to the plant, in that case a signaling molecule is produced, and that signaling molecule will initiate a cascade. Okay, and that cascade will lead to synthesis of some specific secondary metabolites. Okay, now this cascade is actually activating what? A pathway where some of the intermediates of your primary metabolites, they are converted into the secondary metabolite, right? And these secondary metabolites then will help the plant to combat the attack. Now again, within this active response you may have two types. One is called as an active but a local, and the other one is the active but systematic. Means that if a particular plant is injured, okay, and the plant want to overcome that attack in that particular leaf only, so at that particular point only the secondary metabolite is produced and the attack is being combated. But in other case, that if there is an attack onto a leaf, so to prevent the whole plant, what is happening? The signaling molecule is activating the pathway in that particular leaf also, but it is transported to the other leaves or the other parts of your plant where there also the pathway is being activated, the secondary metabolites are produced, and then the plant can be prevented from this different type of the attacks. Right. So this is the type of mechanism which is present into the plant system whenever there is an attack or any kind of disease condition might exist into the plants. Okay, so to continue, I am now presenting the screen, and then we'll see what are the next points which can be covered under this topic. Is the screen visible?
Yes, sir.
Okay. Yeah, so this is what I was talking about, and I think all of you got an idea about this. The two types, that is the passive and the active. Now, to understand that how do the plants they protect themselves, first we should have some basic knowledge that what could be the different type of the attacks which could be there. Okay, so if it is a bacterial infection, if it is a viral infection, or if it is a fungal infection, that can be through the injury or the wound which has happened to the plant, and through that there could be an entry. जसं ह्युमन बिंग्स मध्ये होतं, आपल्या एखादी जखम झाली की त्यातनं देर आर चान्सेस दॅट यू मे हॅव द बॅक्टेरियल इन्फेक्शन or the fungal infection. The same case could be for your plants also. But here in case of the plant, there are some animals or some insects which are actually eating up those particular plants. So in that also there are different categories. So just to have a feel and to have a basic knowledge about that, I'm trying to tell you about these different categories of the insect herbivores which feed upon these plants for their growth, okay, or for their food. So the first category could be your phloem feeders. Means they are not actually creating an injury to the plant, but simply they have a very long beak like structure where they can pierce into the plant directly into the phloem, and from the phloem it will suck up the liquid. Okay, and this under this particular category it comes the aphids or the white flies, the white flies which I told you, which are present nowadays. If you see some of the vegetable plants like मेथीचं असेल किंवा मिरचीची पानं बघा तुम्ही, टोमॅटोची पानं, त्यांच्या खाली बघा एक असे पांढऱ्या रंगाचे तुम्हाला स्मॉल फ्लाईज दिसतील. सो दे आर नथिंग बट दी व्हाईट फ्लाईज or aphids. Now, such kind of phloem feeders, they may cause a little damage to the epidermis and the mesophyll cells, but there is no a vast amount of damage. Then you have the second category, which tells about the cell content feeders. So under this, you may have the insects like mites and the thrips. Now, what do? They pierce or they suck. Okay, they are called as the piercing or the sucking insects. Okay, and such kind of insects, they may cause an intermediate amount of physical damage to the plant cells. Fine. So I think now you got it. Whatever that is, some intermediate amount, some amount of breakage or damage to the cells could be there with the help of this mites and the thrips. And the third one is the chewing insects. Like there are some caterpillars, the grasshoppers, the beetles. So you might have seen that whenever such kind of attack is there of the caterpillar, they eat up those particular plant leaves. Okay, so you might have seen some of the leaves, they have been half eaten or something like that. So slowly they start eating all the leaves, and the plant may die. So these chewing insects are the one which cause the most significant damage to the plants. Now, depending upon that what kind of herbivore or the insect it has attacked that particular plant, you can now imagine what kind of damage could be there. Okay, and depending on that, you can then imagine that what kind of infections could the plant may have. Okay, so this is just for the information which I have tried to tell you. Yeah, so the structural defense or the physical defense which I told you, so I think so on all of you might have studied about the anatomical features of those particular leaf or the stem. So you can see here, so these are the ingredients within the plants. So this is the vacuole, then you have the tonoplasm, the plasma membrane, then you have the middle lamella, above that you have a cell wall. So you have such a characteristic structure of these plant cells, but above that you have a cuticular layer, Above that you have the cuticle proper, okay, and above that what I told you about the physical defense mechanism could be the presence of the surface wax, right? So this such kind of defense mechanism is present into the plant cells. But still there are many insects or many other type of the animals are there which can overcome this particular defense mechanism, and they can cause a huge damage to the plant cells. So the surface wax deposits, you can see it onto the cabbage. Okay, कोबीवर बघा तो एक slimy असतो, it's not slimy but असं थोडंसं kind smooth असतं. So it is a smooth type of a surface. So that is nothing but an surface wax deposits which could be present. So these are the SEM pictures. So you can see such kind of waxy deposits are there onto the leaves, which helps the plant to protect themselves. Now these layers could be made up of some specific uh, compounds like cutin, or it could be the waxes or the suberin. So I told you about these three compounds which are present onto them. So these are some of the examples or the structures of this. So cutin is nothing but the hydroxy fatty acids that polymerize to make cutin. Okay. So simply write down these one sentences. That is what is cutin. So it is a hydroxy fatty acid that polymerize to make the cutin. Wax. So these are the simple straight chain alkanes, or it could be fatty acid esters, it could be the long chain fatty acids, the long chain alcohols, etc. Suberin is what? It is a hydroxy fatty acid that polymerize along with each other to make the suberin. So simply the joining of these long chain fatty acids or the polymerization of them may result into the formation of this suberin. Right? So these are the three major structures in the physical structure. Then how does this bacteria or the other type of the parasitic or the fungus, they infect these particular plants? So their entry, as I told you, it could be through the mechanical wounds. Okay, so many of these insects, they have an ability to overcome the external defense mechanism. They can penetrate through it and then cause the damage. So basically for the microbial entry, the mechanical wound should be there. So this bacteria can cause the damage, okay, because they provide the sites for ice nucleation. Then parasitic nematodes use their sharp mouth part to get through the plant cell walls. So these are the different entry points, what you can say. Some of them, they form the tumors on the roots, what we know in case of the bacteria, agrobacterium or the rhizogens. So they form some tumors. Okay, so that's an actual a disease. So this is how you can now imagine that. Suppose this is a nematode. Nematode, they have a very sharp teeth like structure, so they can easily break that external defense barriers, okay, and they can have an entry. While this is the root which is showing you some of the nodules which are formed, so they are nothing but the infection of the bacteria. Now how does this fungus it can enter? So this is these are the different phases which has been shown that how the actual growth can take place. So obviously there has to be an wound, and wherever there is an wound, a small damage onto the plant parts, then there could be an entry of this fungus. Now as we know that this fungus, they can produce the spores, and these spores, they are so light in weight that they can easily be carried with the air. So these spores, they can land onto those particular leaves where the damage is there. Once it has landed there, it will the spores will germinate. They will form a structure called as an adhesion pad. So that acts as an nucleus from where the hyphae can be generated. So once the adhesion pad is formed, the hyphae will start growth. The hyphae will increase, and slowly it will spread along the cells. And once it has spread along the cells, it will try to penetrate into the cells. Okay, and then the cell damage can be caused. Right? So this is what actually happens, that how does the fungus they can enter. So I think through this image it will be clear. So if there is a small damage, what can happen? the spores can come and sit onto that particular damaged site, so it forms such kind of structure. This is called as an adhesion pad, and through this adhesion pad the germination will take place. So you can see that these hyphae are produced. So this hyphae will enter it through the stomata, and it will try to enter through the different parts of the leaves. Okay, and you can see here that these what you can say, the small hyphae, which are called as the hostorium, they will enter into the cell and they will try to take up the nutrients, and this is slowly it will go on damaging all the cells of that particular leaf. So for such kind of attacks, there are some preformed biochemical defense mechanisms. Okay. Now here some of the sentences might be repetition, but this is just to give the information. uh, so already I have given you an idea about these different type of the sentences. Yeah, I'll just talk about this last sentence, that these secondary metabolite, they display many antimicrobial activity, that is they play an important role in the defense mechanism. So they protect against the herbivory or the pathogens, they act as an attractants for pollinators or the seed dispersing animals, they act as an agents for plant-plant competition or the plant microbe symbiosis. So this picture will give you some idea that what are the different factors which might be affecting a plant and which type of compounds are involved into combating that particular effect. Say for example, if there is a damage by the UV light, that how does the plant they overcome this damage due to the UV light. So in those particular case, you can see that they are producing some compounds called as anthocyanidins or flavonols or flavones. Okay, so these specific compounds are produced, and this compound they prevent against the UV light. If there is some wound, so in that case there are some specific compounds like chlorogenic acid, coumarins or the lignin, they help in preventing that particular wound formation. Okay, thus in case of human beings, if you have a wound, you have the blood clotting mechanism. So that blood clotting mechanism, it comes in picture, and they will form a layer there, and it will prevent the entry of the different bacteria or the other type of the fungus or something like that. Right? Or in many cases, you might have seen that whenever you try to cause a wound onto the plant, they release some sticky material, and that is nothing but the gum what you see. So that's a mechanism which you can imagine, which is similar to that of the blood clotting mechanism. So they cover up that particular wound surface area so that there will be no entry of any foreign material. Then these are the two examples which you can take, which happens above the earth surface. But what happens below the earth surface? Because we know that the soil is a rich source of microorganisms, so there are maximum chances that these plants may have some infections uh, through the bacteria. But at the same time, if the plant has to take up some micronutrients from the soil, they will require some help of the microorganism. Okay, so in the initial lectures I told you, so just to attract this beneficial microorganism, or to what you can say, avoid those harmful bacteria, these secondary metabolites are produced. So if there is some pathogenic attack, so to avoid that particular pathogenic attack, you can say that there are compounds like isoflavones, coumarins, flavonols, or the stilbenes. So these specific compounds are produced which will not allow the pathogens to attack the roots. Okay. So if there is a low iron content into the plant, the iron cannot be easily taken up by the roots from the soil. So it has to be made available. So to attract those specific organisms which sequester the iron, they release the compounds called as phenolic acids. So these phenolic acids, they will attract those microorganisms, they will come near the roots, they will provide the iron near the roots, and the roots can easily take up those iron. The same thing for your low nitrogen. Okay, so to attract the nitrogen fixing bacteria, they produce the compound called as the flavonoids or the isoflavonoids. Then for in case of the low phosphate, you have some compounds called as anthocyanidins which are produced. Right? So whatever may be the case, whether it is a beneficial thing or an harmful thing, so in both the cases, the secondary metabolites are produced and the appropriate function can be taking place into the plants. Okay, so this picture might give you an overall idea. So I think you might have written down some of these things. So the three groups which I have already told you. So the nitrogen containing compound, that is, they are the alkaloids, phenolic compounds, and the terpenes. So within this, you have these different type of the compounds. Some examples which I have quoted here. So in case of alkaloids, it could be nicotine, morphine, cocaine, caffeine, or the glucosinolates. Phenolic compounds, you have the flavonoids, anthocyanins, salicylic acid, terpenes, you have the limenoids, saponin, spinin, and etc. etc. Now the next thing comes that how these secondary metabolites are synthesized. So as I told you that there is a linkage between the primary metabolic pathway and the secondary metabolic pathways. Means that there are some specific intermediates of the primary metabolic pathways which may act as an starting point for the synthesis of your secondary metabolic pathway. Right. So this slide will give an idea that what actually is happening. So this pink color what you see, that is giving us an idea about the primary metabolism. So there you can see that through the process of photosynthesis, this carbon dioxide is fixed. Okay, and that carbon dioxide which is fixed, it will undergo the different pathways. So knowing the compounds, you can imagine what type of pathway it is undergoing. Like 3-phosphoglycerate means it is undergoing the synthesis of the carbohydrates. Then erythros 4-phosphate could be similar to that of the pentose phosphate pathway. Then phosphoenol pyruvate, or the pyruvate means the glucose is being metabolized. It will undergo the TCA cycle. It will undergo the acetyl CoA, formation of the acetyl CoA, and the different pathways are present here. Okay, but then how this secondary metabolites are produced? So these compounds which I have read out, so those compounds, they act as the precursor. Like erythros 4-phosphate, which is an intermediate of your pentose phosphate pathway, can be utilized. Then 3-phosphoglycerate, pyruvate, acetyl CoA, the phosphoenol pyruvate, the some of the amino acids which are produced into the primary metabolic pathways. These act as the uh, points where they can be the precursor for the synthesis of the different secondary metabolites. So for that you have some specific pathways which we are going to see, like you have the shikimic acid pathway, you have the malonic acid pathway, then you have the mevalonic acid pathway or the MEP pathway. So these pathways, they are specifically known to produce the secondary metabolites. Okay, like the shikimic acid pathway will form the alkaloids, that is the nitrogen containing compounds. Okay, few of these pathways can go for the synthesis of the phenolic compounds, or you have another pathway called as the malonic acid pathway. When you talk about the terpenes or the terpenoids, you have these two specific pathways, that is the mevalonic acid pathway and the MEP pathway. Right? So these are the four major pathways which we are going to see, or which you should be aware of to understand that how the secondary metabolites are synthesized from the primary metabolites. Okay, then so the terpenoids. So if you remember, I told you that these terpenoids, they are what you can say hydrophobic in nature, or they are made up of the hydrocarbons. So they obviously, so they should be containing a chain of carbon which is in the reduced form. So there uh, you should keep in mind that these terpenoids, they contain the carbon unit that is called as an isoprene unit, which is a C5 carbon unit called as an isoprene. Okay, so it is called as an hemiterpenes. So like this, these isoprene, they will combine with each other and they may form the different type of the terpenoid. So you can see here C10 means monoterpenes. Okay, so limonene, menthol, it is a C10 compound. Then you have the C15, that is the sesquiterpenes. They are called as the sesquiterpenes. Means three isoprene units are attached to each other and they are forming this compound. The one of the example could be your abscisic acid. Then you have four isoprene which can be joined, so it is called as a diterpenes, that is the C20. Example is phytol, retinol, that is a vitamin A. Then you have the C30, that they are called as the triterpenes. So you can see the structures. Okay, and the example here is the cholesterol, which is usually formed in the human beings, and gibberellin. Okay, in case of the plant system. So these are some of the examples or the classification of the terpenoids. Now, what are the different roles of these terpenoids? So here, see, I am trying to give you some glimpses of the secondary metabolites. So monoterpenes, they serve as a defense against the insect. So I think looking at the images, you may now come to know that what kind of compound they might be having, or the defense what they are helping out. So in case of this, it's a lemon class okay, of fruits. So they produce a compound that is called as lemonine. Lemonine is a monoterpene. Okay, and this monoterpene, it helps in prevention against the insects. So you can write down simultaneously these examples. Yeah, I think you can identify the leaves. So that's the pudina. Now within this pudina, you have a compound or a terpene, that is the menthol. Okay, and this menthol, it helps in defense against the insects. Now the question might come that where are these terpenoids produced, or where are they stored? So these oils or these terpenoids which are produced by this plant, they are stored into the surface glands. So if you go and observe the pudina leaves, you can see that there are very small hair-like structures which are present onto these leaves. So they are nothing but the glands, and within these glands you have these secondary metabolites or the terpenoids which are stored. Yeah, so those hairs, they look like this. So these are the ACM pictures. Okay, so just imagine these are the structures of those particular hairs. So which could be present on your sunflower. Sunflowerचे leaves वर बघा एक असे पांढरे पांढरे केस दिसतात आपल्याला. So those leaves are nothing but the glands, and within these glands you have these terpenoids or the essential oils which are stored. This is about the peppermint, that is the mentha. Okay, किंवा पुदिनाचं हे असं structure. Few more examples about this. Yeah, so that was, they were the monoterpenes चे examples सांगितले. आता इथं triterpenes. Triterpene मध्ये एक example you can talk about the azadirachtin, which is present into the neem plant. Okay, so neem tree, Azadirachta indica. So some scientific names also you should be able to remember. So Azadirachta indica contain this compound called as azadirachtin. So see, such a complicated structure is there. Okay, and this complicated structure is derived from the isoprene units. So it's a lemonoid, it's a triterpene. Then you have another compound, that is ecdysone. Okay, so this ecdysone is a compound which is present into the fern plant. You might have seen the fern plant, not a plant but a herb. So it has this compound called as an ecdysone. So see, look at the structure. It's a triterpene made up of the isoprene units. Now these terpenes, they are basically released whenever there is an insect attack. Okay, and this insect attack, yeah, one mechanism which I told you, that these plants, they can call upon some beneficial organisms, some insects also, some ज्याला आपण म्हणतो मित्र किडे. Okay, so those beneficial insects can be called upon by these terpenes whenever there is an attack onto them. Right, so a beautiful mechanism is there which I'll try to explain it here. Yeah, so this is a plant. This plant is being attacked by this caterpillar. So this caterpillar is chewing the leaf. Now by chewing the leaf, what it is doing, it is causing a cell damage. So it is causing the damage to the glands also, and if the glands are damaged, now you can say that these terpenes, they are released into the air. These are volatile in nature. So those particular terpenes which are released, they will attract these insects. Okay, so these insects are attracted. So these insects will come, they will sit onto the caterpillar, they will lay their eggs. When the eggs will hatch, the young ones, they will eat up this caterpillar and the caterpillar will die. Okay, so this is one of the mechanism which can be found uh, by the plants. Okay, yeah, so this is what I told you, that the volatile terpenes released from the leaves of a maize plant after attack of a lepidopteron larvae or the aphids, they attract the parasitic wasps that are natural enemies of the lepidopteron larvae. Okay, so these are the different type of the parasitic wasps what you can see here. Okay, हे जे दिसत आहेत तुम्हाला हे सगळे parasitic wasps आहे. Yes, so you can see this mechanism which is shown. I got this image, so I thought I'll show you. So from this you get an idea what I told you. So see, this caterpillar is chewing the plant, so the cells are damaged, so the terpenes are released. They are volatile in nature, so this volatile signal, it will attract this insect. This insect will come, it will sit onto the caterpillar. See, they have laid the eggs, the larvae are there, so the larvae, they will grow and they will eat up that particular caterpillar, right? And this is how the defense mechanism is being uh, found in the plant system. It's not all, but many of these plants, they can show such kind of mechanism. Yeah, one more good example. Have you heard about Kaskuta? Kaskuta ऐकलंय का कोणी? बघितलंय का? Am I audible?
Yes, sir.
So have you seen the Kaskuta?
No, sir.
You have not heard about it?
Uh, I don't remember how it looks. Just know the name.
Okay. Has anyone seen Kaskuta? How it is? Okay, actually it's a parasitic plant, you can say. It does not have roots, it does not have leaves, it does not have no, stems it can have, but there are no leaves, no roots, but still it can easily grow onto the other plants. Okay, so it's a parasite, you can say, but this parasite, it can grow onto the plants with the help of the secondary metabolites which are produced by them. Okay, so it's yellow in color. You might have seen that in some plants you have those thread like structure which are yellow in color. I'll show you the image, then you will come to know. तुम्ही कॅम्पस मध्ये आलात तर इथं जे विद्याप्रतिष्ठानचं सेंट्रल ऑफिस आहे त्याच्या समोरच्या झाडांवरती बघा, यू हॅव ह्यूज अमाउंट ऑफ दिस कस्कुटा विच इज ग्रोइंग ऑन टू द प्लांट्स. Okay, yeah, see here this yellow color wave like structure what you can see, so it has spread out throughout the leaves. Okay, so this yellow you can see, so it is simply excuse me, what you can say, yellow thread like structure which does not have the roots, which does not have leaves, but still it can easily spread upon the plants. Okay, so this spreading of this cuscuta, it is guided through the volatiles or some terpenoids which are produced by that particular plant. Means in this case what you can do, you can simply break this cuscuta, मी त्याला थोडासा तुकडा घ्या, and you simply put it onto a plant where you want it to grow. Once you have put it there, what will happen? It will immediately start growing onto that particular plant. Okay, and it will spread throughout the plant within some 8 to 10 days. Easily grow onto that. Now within this also, this cuscuta, there are different types. Means there could be varieties. जसं प्लांटच्या varieties असतात तसे कस्कुटाच्या varieties असतात. So there are two varieties what scientists have studied. So they can easily distinguish the host. काही असतील ते host specific असतील. That they cannot grow on each and every plant. So they will grow only on some specific plants. Okay, so that kind of variation has also been found. So could be that these different types of the cuscuta, they might be producing some different type of the volatiles, and which is guiding them to grow onto the plants. Fine. So this is one very good example what we can quote here, that the growth of this cuscuta. Fine. So that was some of the examples of the terpenoids. Now we'll move to the some examples of the nitrogen containing compound, that is the alkaloids. So in this you have these glucosinolates, one of the best example which can be quoted. So these glucosinolates, they are found into the mustard oils, and they are also what you can say, found in the family, that is the Brassiaceae family, that is the cabbage family. Okay, so these what you can say, this compound, that is the glucosinolates, once they enter into the gut of that particular insect who is eating these particular plants of the Brassiaceae family, what happens? The enzyme called as the myrosinase, it it breaks down the glucosinolates. Okay, and it releases some defense compounds. Okay, and those defense compounds will kill that particular insect. Yeah, before that I have just tried to show you the cabbage family. Can you identify which type of things are shown here? What is this? This image which I am showing with the cursor. Can you see the cursor?
Yes, sir.
Yeah, I'm showing the laser pointer. What is this?
Broccoli,
Okay, good. This is broccoli. What is this?
cauliflower,
Good. These two images are basically this patta gobi ज्याला आपण म्हणतो.
cabbage,
Yeah, cabbage. This beetroot.
mm.
Okay, this is also of the same family. Cabbage सारखं
Garlic.
दिसतं. Garlic नाहीये ते, पण cabbage सारखंच आहे.
अच्छा.
This is also cabbage. What is this?
No idea, sir.
Can anyone identify it? From now onwards, a picture lakshat ठेवा. It's a very important plant for us. It's not actually a plant, it is a grass, but it is very important because it is one of the model organism to study the plant system. What is the model organism? याच्यात खूप काम झालेलं आहे.
Arabidopsis thaliana.
Yes, so it is Arabidopsis thaliana, and it also comes under this cabbage family. Okay, so it looks like this. It's a grass. Fine. देन कॅबेज फॅमिली तुम्ही बघितली, त्या कॅबेज फॅमिलीवरती वेगवेगळ्या प्रकारचे इन्सेक्ट्स, खूप जास्त इन्सेक्ट्स किंवा कॅटरपिलर्स तुम्हाला सापडतील. So this is just to show you the different type of the insects which could be present. So it could be the aphids, it could be the looper. Okay, so they are feeding onto this cabbage family. जरी कॅबेज मध्ये एवढं टॉक्सिक कंपाऊंड्स असतात, तरी खूप अटॅक त्याच्यावरती होऊ शकतो, बिकॉज द इन्सेक्ट्स हॅज ऑल्सो इव्हॉल्व्ड. They also made some modifications within them to overcome those toxic compounds. Right? So there is a defense mechanism that is called as an myrosinase glucosinolate system. Okay, and this is found specifically only into the cabbage family. So how this defense it acts, or how it takes place? See here. So you have these glucosinolate compounds, you have the myrosinase enzyme. So this myrosinase enzyme will act upon the glucosinolates. Glucosinolates will be converted into some products, and these products are the toxic products which will kill that particular caterpillar. Okay, so how this mechanism it happens? So see, through this, although you may see it some complicated, but just try to understand. So within the leaf, Laksha diya. So within the leaf there are two types of cells. Okay, one cell is called as a myrosin cell. Okay, and the other cell is called as an S cells. Okay, so there are two types of cells, that is myrosin cells and the S cells. It is present into the leaf. Now these myrosin cells, they have an enzyme called as myrosinase which is stored into it. Okay, myrosinase, it is called as beta thioglucoside glucohydrolase. Fine. So this enzyme is stored into the myrosin cells. On the other hand, you have the S cells, and within these S cells, you have these glucosinolates, which are stored into the vacuoles of that particular cell. Okay, so these are the sulfur containing compounds which are stored into the vacuoles of the S cells. Now within the leaf, both these compounds, they are stored in the different cells. So there is no reaction which will take place and the plant is safe. But the moment there is an attack of the caterpillar, so the caterpillar will try to chew up the leaves. Now due to that chewing, what is happening? The myrosin cells will be damaged, the S cells will be damaged. So both this compound, that is the enzyme and the glucosinolates, they are released. Once they are released or once they come in contact with each other, what will happen? This myrosinase, it will act upon the glucosinolate and it will cause the hydrolysis of the glucosinolates. And due to the hydrolysis of the glucosinolates, it will be converted into some products. And what are those products? So here you can see that this is an intermediate which is shown, but the products which could be formed could be your isothiocyanate, nitrile, or the thiocyanate. And the glucose is released. Now this cyanate, it's a cyanide type of an toxic compound. So once this isothiocyanate is formed, it will be a toxic compound for the caterpillar. And the caterpillar, if it consumes these particular leaves containing the isothiocyanate, the caterpillar will be killed. Right? So this is how a beautiful mechanism is there into the cabbage family specifically, and it is called as a myrosinase glucosinolate system. I think it is clear. Okay, so this, you have these two cells having these two compounds. Unless and until they are damaged, it's not effective. But once it is damaged, the enzyme will act upon the compound. It will cause the hydrolysis of that compound, and these products which are formed, that is the isothiocyanate, nitrile, or the thiocyanate, they will act as a toxic compound for killing that particular insect, and specifically the caterpillars. Right. So this is one of the insect mechanism what you can see here. Right. So that were those were the few examples of the alkaloids. Now we'll move to the next class, that is the phenolic compounds. Okay, so phenolic compounds, they are the flavonoids. So already I told you why they are called as phenolic compounds, because they contain an aromatic ring with a hydroxyl group. So there are more than 10,000 compounds which are known. Some of them they are soluble, some of them they are insoluble, some of them they form the polymers. Okay, so variety of compounds are there under these phenolic compounds. Now they have a diverse functions like defense against the herbivorous or the pathogens. They may provide the mechanical support. So there I told you about the lignin. Then they can be used for attracting the pollinators. They can help the plant to protect against the UV radiation. They can also help to reduce the growth of the nearby competing plants. Right. So here majority of the uh, ecological functions what we are talking about, that is the interaction of the plants with the other life forms, here this phenolic compound they play a very important role. Then these plant phenolics, they are biosynthesized by many different type of the pathways. So one of the pathway what major basically we are going to talk about, it could be your shikimic pathway or the malonic acid pathway. Right? It's a minor pathway, but the major one is the shikimic acid pathway. Now actually this shikimic acid pathway is found in each and every plant, and basically this pathway is responsible for the synthesis of the aromatic amino acids. But the alkaloids are those compounds which contain a ring structure. So for that you require that ring-like structures, and these ring-like structures are found into the aromatic amino acids. So see how beautifully nature has selected this pathway that is called as an shikimic acid as pathway, which is forming the phenylalanine or the tyrosine or the tryptophan. So these three aromatic amino acids, they can be diverted towards the synthesis of the what you can say the alkaloids, right? And this shikimic acid, it starts from erythrose 4 phosphate and phosphoenol pyruvic acid, and you can see that it can form the variety of alkaloids. आपण हा pathway बघणारच आहोत डिटेल मध्ये, but here just to give you an idea, this pathway is very important for the alkaloid synthesis. Yeah, so some examples which I have quoted here of the phenolic compounds like caffeic acid, ferulic acid. आता कॅफिक ऍसिड कशात असणार आहे? कॉफी मध्ये. Okay, so caffeic acid and ferulic acid, and they inhibit the growth of neighboring plants, and this mechanism is called as allelopathy. हे लिहून ठेवा. I'm telling you, you write it down because the MCQs can be asked on such type of questions. Okay, so as I told you that if they want the interaction between plant, that some of the plant they do not allow the other plants to grow around them. So in those this compound called as caffeic acid or the ferulic acid which is released, which will inhibit the growth of the other plants, and this mechanism of inhibiting the growth of other plants is called as allelopathy. Okay, so caffeic acid and ferulic acid. Then you have ambelliferon and soralin. So these are the two compounds which come under the category that is coumarins. That is one benzene ring and a C3 compound, okay, which is attached to it. So that is called as an coumarin. So both this compound, that is the soralin and the umbelliferon, they exhibit phototoxicity to the insect herbivores after activation by the UV. And next you can see some example called as vanillin, salicylic acid. Okay, so these are the benzoic acid derivatives where you can see that there is a C6 ring structure and C1, and that is an additional carbon is there in the form of carboxylic acid. Right. So this salicylic acid is a plant growth regulator which is involved in systemic resistance to plant pathogens. So the induced pathway which I am trying to tell you. So this salicylic acid is one of the signaling molecule which is produced whenever there is an attack by an insect or a bacteria, right? So it will initiate a pathway for the synthesis of. So salicylic acid is one example of the phenolics. Now within these phenolics, you have another class of compounds which are called as lignin. So lignin is highly complex phenolic macromolecule. Okay, it is the second most abundant organic molecule. It is present into the cell wall. It helps in supporting the cell wall, the conducting tissues. So it acts as a support system. Okay, and it helps in strengthening the stems and the vascular tissue. It has a protective functions, so it provides the physical toughness which deters the feeding by animals. Means some of the animals, they will avoid eating some specific plants. Why? Because they might be containing the high content of the lignin, and this lignin cannot be easily consumed by those animals, so they will try to avoid. जर हे चावताच येत नाही आपल्याला तर कशाला खायला जायचं, so something like that. Okay, so then the chemical durability makes it indigestible by the herbivorous. Means these herbivores, they do not have specific mechanism where this lignin they cannot be digested. Okay, then this lignification, it blocks the growth of pathogens and is a common response to infection or wounding. So I told you, like what we have the blood clotting mechanism, the same way in case of the plants you have this lignification mechanism. So whenever there is a wound or injury to the plant, so this lignification will take place and it will avoid the growth of the or it will avoid the infection by the bacteria or the fungi. Now within this phenolic compound, you have a very large class of compounds which are called as the flavonoids. So we'll talk something about the flavonoids. Yeah. So maybe with this slide I'll stop because it's we are coming to 11 o'clock. Fine. So this is the basic structure of the flavonoid. So if you look at it here, it consists of two rings, A and B ring, and both these A and B ring, they are joined by a C3 carbon. Okay, so 2, 3, 4 carbons. So these are the three carbon which are joining this A and B. Now the uniqueness of this synthesis of the flavonoid is that it involves two pathways which are required for the synthesis of this basic flavonoid structure. So one of the pathway is your shikimic acid pathway, which will form this B chain. Then you have the malonic acid pathway, which will form the A chain. So both these chains are formed, there is a three carbon compound which will act as a linkage between the A and B. So it forms this third ring structure, which we can call it as a C ring. So this is the basic ring structure which is formed in case of the flavonoids by the different pathways. Fine. So it is one of the largest plant phenolics. So 15 carbons arranged in two aromatic rings connected by a 3 carbon bridge. So this is what are the characteristics what you can remember. Fine. So maybe I'll stop here. Yeah. And tomorrow we'll continue with this. There are a few more things which will be talking about the flavonoids. So maybe I'll complete up with these basic things which are required to understand the secondary metabolites. So maybe after that, maybe if tomorrow I finish it up, or day after tomorrow if I finish it up, so within this 1 or 2 day I will try to finish that presentation, and then we'll start with the your presentations. So those who will be talking about the secondary metabolites. Okay, so those who are talking about alkaloid or flavonoid or terpenoid, they will give the presentation first, and then we'll talk about the extraction method. कारण की माझ्या मते लिस्ट मध्ये पहिले एक्सट्रॅक्शन वाल्यांचं येतंय, so they can talk after we have covered up with the types of the secondary metabolites. So I think it's clear now. Write your roll numbers in the chat box. Yeah, any questions? Sonam Pawar आहे ना?
Oh, yes, sir.
Send me the numbers.
Okay, sir.
Yeah, any questions?
No, sir.
No? Okay. Fine then. So I think today is Saturday, na? Saturday, na?
Yes, sir.
ओके. सो मग आयदर सोमवार मंगळवार लेक्चर घेईल मी, आणि मग त्याच्यानंतर आपण जसं मी आता सांगितलं